Hello, good morning. I am Dr.R.Malarvizhi, Associate Professor, PG and Research Department of Chemistry, Sithalakshmi Ramasamy College, Tiruchirappalli. I'm, Today I am going to discuss about clinical trials which are involved in the drug design. Drug design means we have to find out a new drug for new diseases, etc. It is performed in people that are at evaluating medical, surgical, behavioral intervention. It is the primary way to find out if a new treatment like a new drug or diet is safe and effective in people. Clinical trials are aimed on new drugs to find out its therapeutic effect and to ensure that they have no unacceptable side effects. It is very interesting to see that in medicine for the year 1901, the Nobel Prize was awarded to Emil Adolf von Behring for his work on serum therapy, especially its application against diphtheria, which has produced a victorious weapon against illness and health. Another important Nobel Prize for the year 2021 was jointly awarded to David Julius and Arden Petaposhan for the discovery of receptors for temperature and touch. Clinical trials involves various steps. In clinical trials, we have four phases. In the first phase, phase 1, clinical trials are usually carried out on healthy human volunteers in order to evaluate the dose levels to be administered for each of the disease and its pharmacokinetics, which means that what is the effect of the environments inside the stomach and how the drugs are changed inside the pH levels inside the stomach at different pH levels that is given as pharmacokinetics of the drugs and its safety. These things we have to measure in order to find out a new drug. Effectiveness of the drug is not taken into consideration in this phase. During this study, the volunteers are not allowed to take caffeine, alcohol and cigarettes because these things also interact with the drugs. The dose levels that can be tolerated are usually studied first. It is very important to find out the dose. That means what milligram or grams of a drug per kg of the body weight should be administered for each of the disease. For each dose level, we have to take 6 to 12 persons and they are given the active drug and about 4 to 8 persons are given the placebo. Placebo means the compound or chemicals which does not produce any medicinal activity, inert compound. The initial dose level is based on the toxicology results. That means if the drug produces any unwanted side effects, that is toxic. So the dose level is based on the toxic effect of the drug, new drug, from animal testing. First, in order to find out the toxic effect, the drug is given to animals and finding out the potency of the drug as well as the toxic effect of the drug. For example, if the higher dose found to have no toxic effect in animals is, suppose if it is 50 milligram per kg, then the initial dose level will be 1/10 of this weight, this value. After the administration, pharmacokinetic studies are carried out. If the drug is safe, then a higher dose will be given and this will be continued until mild adverse effects are observed. This is taken as the maximum tolerated dose. Studies are also carried out to determine whether there are any interaction between the drug and the food. This study is essential in order to establish when the dose will be given relative to the meals. Special studies are also have to be carried out on volunteers with renal or hepatic impairment and gastrointestinal problems. In this case, bioequivalent studies are also required when a drug is to be taken in different formulations, that is in tablet form and syrup form, um, and injection form, etc. It has to be demonstrated that dissolution of both formulation is similar. In phase 1 study, it takes about a year and involves about 100 to 200 volunteers at the cost of 2.5 to 3 million dollars. In phase 1 clinical trials, there are different steps involved: a single ascending dose, multiple ascending dose, and food effect. In phase 2 clinical trials, generally it has about 2 years to complete, and it is started before the completion of the first phase. These studies are carried out on patients, whereas in phase 1 we have used healthy human volunteers. In phase 2, patients are used for the study trials. Our aim is to establish the therapeutic value of the new drug and to study the pharmacokinetics again and the short-term safety of the drug. These are also used to establish the best dosing regimes, though dose of the drug should be found out in this case. Phase 2 studies are divided into two types again, that is early studies and later studies. Initial trials, early studies are also known as initial trials, that is named as 2A, involve a limited number of patients to see whether the drug has any therapeutic value and in order to find out whether it has any side effects. Later studies involve a large number of patients. They are usually carried out as double-blind placebo-controlled studies. That means double-blind means in this study both there are two groups of patients. One group is given the drug, actual drug, and the other group is given the placebo. The placebo means, again I will tell you, it is nothing but the inert material which does not produce any medicinal activity. A double-blind study means that neither the doctor nor the patient knows whether the placebo or drug is given to them. Studies may be carried out on special age group of volunteers. For example, the drugs intended for Alzheimer's disease is tested on healthy elderly volunteers. The studies demonstrate whether the health of patient group receiving the drug is improved relative to the patient group receiving the placebo. The studies also explore different dosing levels and regimes to try and clarify which regimes are the most effective. For patients suffering from cancer or AIDS, it is unethical to use patients to give placebo or keep the patients in placebo. So an established drug is used as standard, which is compared with the new drug for studying. In order to find out the new drug for cancer or AIDS, we should not use placebo. It is unethical to use placebo for patients suffering from cancer or AIDS, so that a standard drug is used, which is the activity of the standard drug is compared with the new drug for these types of diseases. In phase 3, this study includes fewer dose levels and more patients per dose in order to show statistically significant efficacy and safety. In phase 3, efficacy and safety is to be find out. So fewer dose levels and more patients are involved. In phase 3A trials are carried out prior to the drug's registration with the regulatory authority, whereas Phase 3B trials are carried out after registration but prior to the approval. These studies are carried out in order to compare a new drug with other drugs already established in the field. Phase 3 studies can be divided into two types again. Phase 3A, Phase 3B may begin before the end of the Phase 2. There are two types of Phase 3A and Phase 3B. Phase 3A involves testing different stages of disease indication, dosage forms, formulation of drug, different routes of administration, for pharmacoeconomic evaluation. And in Phase 3B, the trials will continue while the regulatory submission is pending at the appropriate regulatory agency. This allows the patients to continue the same drug and in phase 4 for patients suffering from cancer or AIDS, it is unethical to keep the patients in placebo. So an established drug is used as standard, which is compared with the new drug developed for the cancer or AIDS. In phase 4 studies are performed after the regulatory authorities have granted market approval for the drug. These studies document the safety of the drug and identify the unexpected interaction with the other drugs. They also assess the drug's therapeutic value in the special populations like elderly patients suffering from Alzheimer's disease and the patients suffering from cancer or AIDS, etc. On an average, for every 1,000 structures synthesized during drug design, 500 drugs only reach for animal testing, and from these 500 drugs only 100 reach phase 1 clinical trials. From these 100 drugs only one drug is go to the market as a drug. The average overall development cost of a drug is estimated as being $114 million in 1987. On an average, for every 10,000 structures synthesized during the drug design, only 500 drugs reach for animal testing. Among these 500 drugs, 100 reach phase 1 clinical trials. From these 100 drugs, only one go to the market. The average overall development cost of the new drug was estimated as being $114 million in 1987. A new study in 2020 estimated that the median cost of getting a new drug into the market was $985 million and the average cost was $1.3 billion, which was much lower compared to previous studies, which have placed the average cost of drug development as $2.8 billion. Based on data, 63 therapeutic agents have developed by 47 companies between 2009 and 2018. Thank you.